In this video, we're going to walk through the answers for the in-class worksheet for Chapter 3, which also includes some material from Chapter 2. So, Part 1 is a video about the inner life of a cell. So I've posted a video representing a virtual tour inside of a white blood cell, specifically a neutrophil, as it is attracted to the site of an injury in the body and it immigrates from the bloodstream to the injured tissue. You'll learn more about this process in Biology 2252. First, I suggest watching the video in its entirety and then re-watch clips as you attempt to answer the questions. The clip times are labeled in the video on D2L. You can find the video in the Kaltura Media Gallery on D2L. So hopefully you've already watched the video in its entirety. This is one of my favorite videos ever made. I will show you the clips as we answer the questions, but if you haven't watched the video in its entirety yet, I suggest you pause this video, go watch the video in its entirety, and then come back. Alright, so we're going to take a look at clip 1 to start with. I've pulled up the video in the Kaltura Media Gallery, and for those of you that don't know, you've got the three little lines up here in the upper left that allow you to navigate around from one clip to another, or for the lecture videos from one section to another. I've also turned the sound off for this particular lesson so that I can point out certain things to you. So we're going to start with clip 1. So what we're seeing here is the inside of a blood vessel. Those are red blood cells flying by. This is a white blood cell, so that's the neutrophil that we're about to go deep inside. So now we are looking at a close-up view of some of the proteins. The purple molecules are on the neutrophil. The yellow molecules are on the cells that line the blood vessel. And so what you're seeing is that this white blood cell is slowing down and is actually anchoring itself to the wall of the blood vessel. So at the bottom we have the white blood cell. At the top we have an endothelial cell lining the blood vessel. So that was the end of clip 1, and so now we're going to answer some questions together. So again, that clip was showing a white blood cell moving through the bloodstream, and we were able to see some of the proteins that were sticking off of the surface. So the first question is, what are the 6 functional types of membrane proteins? So remember, proteins that are in the membrane can serve as anchoring proteins to anchor a cell to another cell or to another structure, like the extracellular fibers. They can act as recognition proteins. These are usually glycoproteins or proteins with little sugar stuck on them. They can act as enzymes and help catalyze reactions. They can act as receptor proteins where something is specific for binding to them. Or they can be carriers or channel proteins and allow substances to pass through the membrane in a very specific manner. So question 2, what type of molecules do you think the purple and yellow molecules are? Why do you think that? What do you think their function is? So here is a screenshot from the video. So again, we had the white blood cell at the bottom with the purple molecules coming out, and we actually had a cell that is lining the blood vessel called an endothelial cell with the yellow molecules sticking out. And so if you guessed that these molecules, the purple and yellow molecules, are proteins, you would be correct. And we assume that they are proteins, membrane proteins in particular, because they are sticking out from the plasma membrane. So why do you think that? Because they're sticking out from the plasma membrane. What do you think their function is? These look like they could be anchoring proteins that are anchoring the white blood cell to the wall of the blood vessel. Okay, question 3. What is meant by specificity? How can you apply this concept to what is happening in this clip? So we saw a close-up view of the purple molecules interacting with the yellow molecules. And the idea of specificity is that most membrane proteins are very specific for the solute or ligand they interact with. So in this clip, these purple molecules are likely to only interact with the yellow molecules. And so these two proteins would be specific to one another in the way that a specific key fits a lock. And so you can see how even the shapes are a bit complementary, where the purple molecule has little extensions that help it to hold on to the end of the yellow molecule. A fun fact, these compounds are called P-selectins, and they are actually cell adhesion molecules that endothelial cells, again, those are the cells that line the blood vessels, put on their surface to attract white blood cells to the site of an injury. So a little fun fact for you. You don't need to remember that. Okay, let's watch clip 2. We left off at the start of clip 2, so I'll just pick up here. So we see all of these little molecules. I want you to notice how everything is moving, right? Nothing is sitting still. Everything is constantly in motion. We like to think of cells as very static because that's the way they look in textbooks. But in reality, things are always moving. You see the little tails in here? I love the way this one shows the tails. The little heads, that gives you a hint as to what these molecules are. And that is the end of clip 2. Okay, so question 4 says, what are the small teal, green, and blue molecules in this clip, and describe their structure? So here is a screenshot from the video. So we are talking about these little small teal, green, and blue molecules. And if you guessed that they were phospholipids, you would be correct. So this is the phospholipid bilayer of the plasma membrane. So phospholipids have a hydrophilic head and a hydrophobic tail. They form a bilayer with the heads facing out to the aqueous solution outside the cell and inside the cell, and the tails sandwiched together to form a hydrophobic middle. So in the video, you can see the little heads facing the outside of the cell. You can see the little heads facing the inside of the cell. You can see both the top and the bottom heads have 2 little tails each. If you look closely in the video, you can see they did a really good job. They did only put 2 tails on each phospholipid, and so this portion in the middle would be the hydrophobic middle. All right, and so there are the labels: heads facing extracellular space, heads facing the cytoplasm, and the hydrophobic tails. Number 5, some of these small teal, green, and blue molecules have molecules sticking out from them facing the outside of the cell. What are these types of molecules? What is the whole collection of these molecules called? And what is their collective function? Again, here's a screenshot from the beginning of the clip. So we saw all our little phospholipid heads all around, and then we noticed that some of them had these little molecules sticking out of them. So these are the carbohydrates that are attached to the phospholipids. All of the carbohydrates together on the outside of the cell make up the structure called the glycocalyx. The glycocalyx functions to lubricate, protect, anchor, help with locomotion, provide specificity, and recognition. And the number 6, where is the cholesterol located? What is its function? So again, in the video, they pointed the cholesterol out to us. So if you noticed, the cholesterol is sandwiched between the hydrophobic tails of the phospholipids. You might have also noticed that the cholesterol only existed in certain regions. And if you watch the video carefully, you can notice that these regions are a little bit more stable and move around less than the regions that don't have the cholesterol. So that hints at its function. Cholesterol provides stability to the plasma membrane. Okay, let's watch clip 3. Okay, so we're about to watch clip 3. We've already started 1 second into it, but I'm just going to pick up here. All right, so we see this purple molecule down here is on the cell we are looking at. This is a molecule on the other cell. Notice the lock and key mechanism that just happened right there. And that's it for this one. That's a short clip. So in that clip, they labeled the orange molecule as a proteoglycan, and they labeled that purple molecule as a receptor. So the first question, number 7, what is a proteoglycan? If you remember, a proteoglycan is a protein with a carbohydrate attached to it. These can also be called glycoproteins. And so the protein portion would be this thick orange region. Probably also this yellow area over here looks a bit proteinish, and the carbohydrate is probably this very thin line right here that's connecting the 2 protein blobs. Number 8, what are some things that you can conclude about this receptor? Well, this goes back to, well, first of all, knowing that the receptor is a membrane protein, it is attached to the membrane, but also remembering that receptors are very specific for the ligands they bind to. So again, we have that idea of a lock and a key, and the little protein blob on the end of this proteoglycan is made perfectly to slot into this receptor. Another molecule would not activate this receptor. And then question number 9, we are looking at an extracellular area in this clip. So that's the space between two cells. So if you remember, we had our white blood cell, and up here we had the endothelial cell lining the capillary. So what can you say about the potassium ion concentration in this area as compared to the inside of the cell? And what about the sodium ion concentration? So remember, these are things that you'll really need to remember, especially for the remainder of the semester. So if we are looking at the outside of the cell, we could say that in the extracellular space, the potassium ion concentration would be lower than inside the cell. So remember, you have higher potassium inside the cell, and right now we're looking at an outside view. Conversely, in this extracellular space, the sodium ion concentration would be higher than inside the cell. So again, inside the cell, sodium is low and potassium is high, and because we're looking at the outside, it's the opposite. Sodium is high and potassium is low. Okay, let's take a look at clip 4. Okay, we're going to watch clip 4 here. So again, what we've actually done in this case, now we're underneath the plasma membrane looking up at it. So it's showing us a couple of membrane proteins. There is one, there is another. Again, I love the way they represented those little tails, the little phospholipid tails. Notice how the heads are moving, and then we're going to stop right there. Okay, so let's look at question number 10. You are shown a large purple molecule and a large molecule with yellow, orange, and light green parts. Both of these molecules represent proteins. Which one is an integral protein? Which one is a peripheral protein? And how are they different? So the purple molecule in the picture at the top here is what we call an integral protein. And it's an integral protein because it completely crosses the plasma membrane from one side to the other. Also, these can be called transmembrane proteins because they go across the membrane. Then this big yellow, orange, green molecule shown down here is a peripheral protein. It's tethered to the plasma membrane, so we can see where it actually has a slight attachment, but the majority of the protein is not actually embedded in the membrane. It just sits very close to the surface. So that's the difference between the two, is that integral proteins are embedded into the membrane, whereas peripheral proteins are associated with the membrane but not embedded into it. So now we're going to watch clip 5. Okay, now we're getting some really cool stuff with the cytoskeleton. So let me go ahead and start playing this. So again, we're on the inside of the cell looking up. That's the plasma membrane up there. So we're pulling away. Notice you've got this skeleton-like structure that serves as a support underneath the plasma membrane. Hint, hint. And then as we get down and further into the cell, again, notice how fluid things are. Things don't just sit there and stay the same. Things are always moving around on the surface of the cell. But as we look down closer into the bottom of the cell, again, something that looks a lot like a skeletal structure. And these are the actin filaments or microfilaments, right? So notice how much they look like scaffolding, and they look like, you know, something that is holding up the cell and giving it shape and structure. And then here's really cool. So this is actually showing the actin filament being formed. So those are little pieces of actin flying to form the actin filament. Again, I love this video because it gives you an idea of how dynamic the inside of the cells actually are. Okay, so we saw an actin filament being made. There are also proteins, like we just saw that little guy called a severing protein. It comes up, it binds, and now the actin filament is being disassembled. And we're going to stop there. Okay, so question number 11 says, you are shown a spectrin tetramer. These molecules interact with actin proteins, which were the little guys shown in purple at the top. What type of cytoskeletal element are we likely looking at? What is the likely function of this part of the cytoskeleton as shown in the video? So if you remember, actin is the main protein component of microfilaments, which are also called actin filaments or thin filaments. You do need to know which protein goes with which part of the cytoskeleton. So actin goes with microfilaments, tubulin goes with microtubules, keratin is frequently found with intermediate filaments, and myosin is found with thick filaments. So the likely function of the cytoskeleton shown in this part of the video is support and anchoring. In fact, actin will often bind to integral proteins in the plasma membrane. So the actin up here, the little purple pieces, can actually bind to some of the membranes that are in the plasma membrane, and that can help anchor the cytoskeleton to the surface of the cell. Number 12 says, next you are shown actin filaments. Notice that the framework of these actin filaments are close to the periphery of the cell. What are the three functions of actin filaments? So the three functions of actin filaments, which again are also called microfilaments or thin filaments, they anchor the cytoskeleton to the integral proteins, as we just talked about. They also determine the density or consistency of the cytoplasm, and they interact with myosin and the thick filaments to produce movement or to change cell shape. And we'll see a lot more about that when we get to the muscle chapter. And then you saw that one view in the video where you could just see how the actin filaments were in there and they were very dense. So you can imagine how the number of microfilaments that you have can determine how dense the cytoplasm is. So if a cell wants to maintain a specific shape indefinitely, it would have a lot of microfilaments to give it a certain shape and density. But if a cell needs to, say, flatten out or squeeze somewhere, then it could reduce the number of microfilaments to give it a bit more flexibility. Number 13 says, note the dynamic nature of the actin filaments. What is meant by dynamic nature? So in the video, we saw how the microfilaments can be quickly assembled. So we saw these little individual actin proteins flying in and coming together to make an assembled microfilament. And then we also saw a little bit later on where the severing protein came along, cut it, and then it started disassembling. So the dynamic nature means that they are constantly changing by being assembled and then disassembled as the cell needs. It's continuously being reorganized, the entire cytoskeletal system. So a lot of times we compare the cell's cytoskeleton to our skeleton, but this is a big difference. You don't break down bones and get rid of them when you want to, say, squeeze through a tight space and then rebuild your bone on the other side. But cells have that capability. Okay, now we're going to watch clip 6, which is one of my favorite. So as you can already guess by the paused picture here, we're going to be taking a closer look at microtubules. So just like the actin filaments, microtubules are dynamic. They can be built, they can be broken down. So here you see the tubulin protein coming together and zipping up. Here's an example of one being disassembled, so it's not needed anymore. So it's breaking back down into individual tubulin proteins. Again, cytoskeleton, very dynamic. Now here's everybody's favorite little guy. This is a little motor protein carrying a vesicle and using a microtubule as a track. And they actually look like this. Scanning electron microscopes have shown that this is what these little motor proteins actually look like. It's like they've got little heads. Oh, I think that's the end of the clip. We'll talk about more of them in a second as we answer questions. Okay, so number 14, describe the structure of a microtubule. What is the primary protein component of microtubules? So microtubules are large hollow tubes formed from a protein called tubulin. And in the video, we saw the microtubules, we saw the individual tubulin proteins that were coming together. We saw the assembled microtubule. Notice how it is larger than that actin filament we saw earlier and how it's hollow. So the tubulin actually comes together and circles around a hollow interior. Number 15, what really cool function of microtubules is being illustrated in this video, hint the part with the vesicle. What type of protein is carrying the vesicle? Is this an active or a passive process? So here's a little screenshot, and yes, I was trying to say all of this in the video, but these little proteins actually do look like this. It's been shown by scanning electron microscopy. So this clip is actually showing the microtubules. If you remember when we went over the functions of microtubules, one of their functions was to serve as tracks for vesicle and organelle movement, and that is what is happening here. So this is like a track, think of it like a train track. And then this little guy is the motor protein. These are also called molecular motors, and two examples are kinesin and dynein. And they actually grab a hold of the vesicles or an organelle, and they move them along these tracks. And they actually do take little steps like this. So each little swing of the foot costs 1 ATP. So it's like they've got little hands up here that hold on to the vesicle or an organelle, and then the little feet take one step at a time along the track, and they use 1 ATP molecule with each step. And you can see in the background, there's another guy back there carrying another vesicle along another track. So this process does require ATP, and that makes it an active process. So I'm about to play clip 7, and this is a very fast clip. So before I hit the play button, we have a nice frozen screen here where you can actually see the microtubules extending throughout the cell. You can see another little vesicle being carried along the one in the bottom right corner, another little vesicle being carried up there. You can see these microfilaments, which are also radiating through the screen. So it gives you an idea of how much is going on inside each of your trillions of cells. Okay, so let's take a look at this short little clip. So this clip is showing us one of the organelles, and it's being moved along these microtubule tracks. And that's it. That was a short clip. So question number 16 was, what organelle is shown in this clip? What is the function of this organelle? And is this a membranous or a non-membranous organelle? So that was a mitochondria. So that kind of told you the answer. So another thing that you could tell if you weren't quite sure that it is a mitochondria is that it has a double membrane. So you can see the membrane on the outside. It's that sort of very light, kind of hazy, lighter color around the outside. And then you can see the inside, the internal membrane, which is folded into these little structures called cristae. And that's part of the structure of a mitochondria. Mitochondria's function is to produce ATP. So they are the energy powerhouses of the cell. And mitochondria are membranous organelles. And again, as I mentioned, they have a double membrane. And you can see that the organelles also move along the microtubule tracks, not just vesicles. Okay, clip 8. Clip 8 is another relatively short clip. So we are going further into the cell. Now we can see the centrosome with the nucleus behind it. You can see a lot of the microtubule tracks and vesicles moving. Still see a mitochondria moving. Okay, so the questions related to clip 8. What is the centrosome? And so it showed you this picture. And so remember that the centrosome is the region of cytoplasm surrounding and containing the centrioles. So it's usually located close to the nucleus. So that's the nucleus in the background. And the centrioles are these two hollow tubes. They actually have a very similar structure to a microtubule. Just like a microtubule is hollow, so are the centrioles. And then again, you can see the microtubules radiating out from the centrosome. Sometimes the centrosome is considered to be the heart of the microtubule skeleton of the cell. Number 18, describe the structure of the centrioles. Name a cell that lacks centrioles. What is the function of a centriole, and what does this mean for the cell lacking a centriole? So centrioles have a 9+0 arrangement of microtubules. That means they have 9 triplets with none in the middle. And cells without centrioles include mature red blood cells, skeletal muscle cells, cardiac muscle cells, and most neurons. Since centrioles are responsible for forming the spindle apparatus during mitosis or cell division, cells without centrioles cannot divide. So your red blood cells can't divide, but they are rapidly replaced with new ones all the time. However, your skeletal muscle cells, cardiac muscle cells, and most neurons also can't divide, and these guys are not replaced. So one important benefit to being healthy and living a healthy lifestyle is taking care of these cells that if you damage them or cause them to die will not be replaced. Okay, last clip. Okay, so clip 9 is the longest one, and while it technically ends before the end, I'm going to go ahead and let this play out. I'm also probably going to give some of the answers away because I want you to see what's happening while it's happening. So we're going all the way to the nucleus now. So this is the nucleus at the bottom. Those are little mRNAs coming out of the pores. The mRNAs come up, they form a special structure. Then look, that's a small subunit that just flew in, a large subunit that just flew in. That is a ribosome. So here's a closer look. Ribosomes are making proteins. Now he's going to let go of the mRNA. Now we have another ribosome making a protein. That is a cytoplasmic protein because it stays in the cytoplasm. But sometimes you can have a ribosome come in and attach, as we see again, small subunit, large subunit. But now it goes and attaches to the rough ER, and it makes the protein and spits it into the rough ER. So I don't know if you just saw him spit the protein in there. Sometimes they can make proteins in the rough ER, and they become membrane proteins. So that was the creation of a membrane protein. So here is our rough ER. We're making changes to the proteins, then we're packaging them in vesicles and sending them on to the Golgi. And of course, vesicles are going to be shuttled around by using these little molecular motor proteins. Just like we saw before, vesicles are made out of membrane, so those are phospholipids. So here are vesicles traveling along tracks to the Golgi. So this is the Golgi apparatus. This is where the proteins are going to be further modified. And then after they pass all the way through, they're going to come out the other end. Again, they're going to be carried along microtubule tracks by motor proteins. Now this vesicle that contains all these nice little proteins is headed up back to the cell surface. So we're going up to the plasma membrane. When we get to the plasma membrane, the vesicle fuses and lets the contents out to the extracellular space. We also add new membrane proteins to the plasma membrane. So this is just a quick little overview of some things we've already seen before. That's about the specificity of a receptor to the ligand it binds to. That was an integral membrane protein. That was a peripheral protein. Again, notice the movement of your phospholipids and their two little tails. We're on the outside of the cell now. So up here, this is the endothelial cell. Down here, this is our white blood cell. These are some other processes going on that are beyond what we're covering in this class. But this is, again, a good idea of showing how membrane proteins can provide anchoring and can allow one cell to adhere to another cell. And what was going on in this entire thing is that this white blood cell was slowing down and getting ready to squeeze out of the blood vessel. So it squeezes between the cells. So these are endothelial cells that line your capillaries. The white blood cell is squeezing between two of these endothelial cells so it can move out into your tissues and chase down any bacteria you've been infected with. So that is actually what the white blood cells do when you have an infection. Like if you get a cut and that cut gets infected, they're going to leave your bloodstream and go into the tissues to fight the infection. But a lot of those processes, even though we were looking at a white blood cell, remember that most of those processes are happening inside of all of your cells. So even when you're sleeping, you've got all of this stuff going on inside of all of your little cells. So hopefully you will never take your cells for granted again. Okay, so now we can finish up the part 1 questions. Number 19, which organelle has the pores as shown in the video? What is a long skinny molecule coming out of the pores? What happened to this molecule in this organelle just before it left through the pores? So as we mentioned when we were watching it, the structure down here at the bottom is the nucleus, and all of these little holes that have proteins surrounding them, these are the nuclear pores. And the mRNA are those long, skinny strands that we saw coming out of the nuclear pores. So remember, inside the nucleus, oh, that's my next bullet point. Inside the nucleus, the mRNA was transcribed using DNA as a template. So remember, transcription is when we're going from this one language to the same language. In this case, we're going from nucleic acid language to nucleic acid language. Then, before the mRNA could leave the nucleus, it underwent some processing where, for example, the introns were cut out and the exons were spliced together. Number 20, describe the structure of the ribosome. What event is being shown with the ribosome? What is the brown curly molecule coming out of the ribosome? So the ribosome is composed of a large subunit and a small subunit. It's also composed of rRNA and protein, so that's another possible answer. So the large subunit was shown in the video, the small subunit was shown in the video. And the event being shown here is translation. So remember, with translation, and then this is the mRNA strand that it's reading. In translation, we are going from mRNA, which is a nucleic acid, to a protein, which is in an amino acid language. So it's going from one language to a different language. The brown molecule being spit out is the protein that is being made. And then number 21, in the video, you see both a free ribosome and a bound ribosome. What is the difference between the two? So in the video, we saw that free ribosome that was just making the protein as it zipped around the mRNA, and then the bound ribosome went over to the ER and attached. So free ribosomes are floating free in the cytoplasm, and they make cytoplasmic proteins. Bound ribosomes are attached to the rough ER, and they make proteins that will be secreted, inserted into the membrane, or that will eventually become part of a lysosome. Number 22, what happens in the rough ER? So we saw the rough ER, we also saw the creation of one of those membrane proteins, and in the rough ER, newly synthesized proteins are modified. For example, sugars can be added to make glycoproteins or proteoglycans. Then they are packaged for transport to the Golgi apparatus. And then what happens in the Golgi apparatus? Proteins are further modified, as are phospholipids, and repackaged to be sent out to one of two destinations. And again, I love how the video is very consistent in even showing you the microtubule tracks along which these vesicles were moving. So even going from like the ER to the Golgi, they had to follow a microtubule track. Number 24, when a vesicle leaves the Golgi apparatus, there are a couple of possible destinations for that vesicle. Describe these two options and then state which one is being shown in the video. So the vesicle can become a lysosome. So if it contains digestive enzymes, it stays in the cell and acts as part of the cell's like stomach or digestive organelle. Or the vesicle can fuse with the plasma membrane, releasing secretory proteins and adding new membrane proteins. In the video, we see the vesicle being moved to the cell surface. And again, we saw the little motor protein carrying it up the microtubule tract, and then we saw it fuse with the plasma membrane and release its contents out to the extracellular space. So what form of cellular transport is being shown at the end? Is this a passive or an active process? So here I did a screenshot where you can see the vesicle is like half fused, and you can see some of the secretory proteins that are being ejected out. So this is called exocytosis, where the vesicle is fusing with the membrane and dumping the cargo out. And vesicular transport, whether it's exocytosis or endocytosis, it's always active. So it's always going to require ATP. Okay, so that was the end of part 1. Part 2 is an organelle review. Not all cells have the same number of each type of organelle. For example, the composition of a neuron would be very different from the composition of an intestinal cell. Below are some examples of how cells can have very specific functions which can determine the number and type of certain organelles to be found in that cell. Below are some descriptions of specific cell types that are found in the human body with some questions for you to answer. After that, we will explore some diseases that involve problems with specific organelles. So this kind of goes to a couple of your learning objectives, including being able to know how the type of organelle abundance might change from one cell type to another. Number 1, a neuron is specialized for sending electrical signals. This requires the movement of sodium and potassium ions across the cell membrane. Thus, a neuron has a lot of sodium-potassium exchange pumps. Briefly describe the function of the sodium-potassium exchange pump. These pumps require a lot of energy in the form of ATP. As a result, what organelle would you expect to be found in abundance in a neuron? So to remind you, the sodium-potassium pump is something that I 99.9% sure will ask you about on the exam, so you should really know how this guy works. So remember, we use one ATP molecule to pump 3 sodiums out of the cell and to pump 2 potassiums into the cell. So 1 ATP for 3 sodiums out and 2 potassiums in. The sodium-potassium pump is essential in order to maintain a concentration gradient. This is how we keep the outside of the cell high in sodium and the inside of the cell high in potassium. So because we're using a lot of ATP, what organelle would you expect to find a lot of? The answer would be mitochondria, because these are the guys that make the ATP that this pump needs to operate. Question 2 says the pancreas has several types of cells, but all of the pancreatic cells specialize in secreting proteins. For example, the acinar cells of the pancreas synthesize and secrete digestive enzymes, while the beta cells synthesize and secrete the hormone insulin, which is also a protein. What type of organelle or organelles would you expect to be in abundance in a pancreatic cell? Since pancreas cells make a lot of proteins, like these enzymes and insulin, there would be a lot of organelles associated with the synthesis of proteins. So think about what organelles are involved in synthesizing proteins. So you would expect to find a lot of ribosomes, a lot of rough ER, and a lot of Golgi apparatus. So again, we are just connecting what type of organelles would help a cell with its major functions. Number 3, the cells of the adrenal gland specialize in producing steroid hormones. For example, the adrenal cortex produces cortisol, a stress hormone made from steroids. What type of organelle would you expect to be in abundance in an adrenal cell? So basically it's asking you, what type of organelle helps you make steroid hormones? And the answer would be the smooth ER. So you would expect to see a lot of smooth ER, since one of its functions is to make steroid hormones. Number 4, macrophages are white blood cells that specialize in hunting down foreign cells and foreign matter within the body. When the macrophage encounters a foreign object, it phagocytizes it. Phagocytosis is a subtype of what type of cellular transport? Is this an active or a passive process? What type of organelle would you expect to find in abundance in a macrophage? So phagocytosis is a type of endocytosis, which is a type of vesicular transport. The other 2 types of endocytosis are pinocytosis and receptor-mediated endocytosis. This would be an active process, so all of the vesicular transports, exocytosis and endocytosis, are all active processes requiring ATP. And you would expect to find a lot of lysosomes to digest the foreign objects that are brought into the cell through phagocytosis. Number 5, I am a cell in the human body with no nucleus and no centrioles. In fact, I have very few organelles at all. What type of cell am I? You would be a red blood cell, or an RBC. So as they mature, they lose their nucleus and most of their other organelles, and they basically just become little sacs full of hemoglobin. And you'll get a lot more next semester when we get to the cardiovascular system about the structure and function of red blood cells. Number 6, Tay-Sachs disease is a type of lysosomal storage disease. This disease causes a progressive deterioration of nerve cells, resulting in severe mental and physical disabilities beginning around 6 months of age. It usually results in death by the age of 4. Using what you know about lysosomes, explain why lysosome dysfunction could cause such a severe disease. So remember, lysosomes are the digestive centers or the trash compactors of the cell. So if a cell uses a lysosome to break down foreign matter, but it also uses lysosomes to break down damaged and worn out cell parts. So if a cell is unable to break down old and damaged organelles and other objects, then these waste products are going to build up inside of the cell, and eventually the cell is going to be so full of waste products it can't do its normal job. So think of what would happen if you never took out your trash, right? It would just keep accumulating and accumulating and accumulating, and eventually you wouldn't even be able to walk around your house. And then we're talking about neurons in this particular type of disease, the Tay-Sachs disease. And so once a neuron can't do its job, the body has lost its main control center. The brain can't function, the nerves can't function. And so this causes serious deficits, and this is why it always ends in death at a very young age. Number 7, Zellweger syndrome is a rare disorder that is characterized by the reduction or absence of functional peroxisomes. What type of molecule do you think builds up in these patients? So what do peroxisomes break down? Peroxisomes break down fatty acids. So this syndrome results in a deadly buildup of fatty acids. This one is actually more lethal more quickly than the Tay-Sachs. Patients actually normally do not survive this one past the age of 1 year old. So without being able to break down those fatty acids, they build up quite rapidly. Number 8, both Alzheimer's disease and Parkinson's disease involve the buildup of proteins in the brain, which form plaques that can cause neuronal damage. What organelle do you think could be a target of research for these diseases? So let's think. Okay, we have diseases where we have too many proteins building up and forming clumps. So what organelle breaks down proteins? So if you said proteasomes, good job. Proteasomes break down proteins, so they could be a target for treating these diseases. So if there was some way we could get the body to tag these excess proteins for destruction, maybe it could be a type of treatment, or, even possibly a cure, which that would be amazing. But in fact, if we get into some of the details of Alzheimer's disease and Parkinson's disease, there's some research that's showing that the reason those proteins actually build up in the first place is that there is a problem with ubiquination. So remember that a protein is tagged with a little tag called a ubiquinin, and this tells the proteasome, hey, this protein needs to be broken down, right? So it's like going around and just tagging stuff for disposal. And so it's thought that in people that have these diseases, one of the problems could be that they are not putting the tags on these proteins like they should, so the proteins are staying around and then clumping together to make these plaques. So part 3 is a diabetes case study. So diabetes affects 25.8 million people in the U.S. The number of young people that are annually diagnosed with diabetes is on the rise, with 15,600 diagnosed with type 1 diabetes and 3,600 diagnosed with type 2 diabetes. In 2007, the estimated annual cost of diabetes was $174 billion. Average medical expenditures of individuals with diabetes are 2.3 times higher than people without diabetes. The major complications with diabetes are heart disease, stroke, high blood pressure, blindness, kidney disease, nervous system damage, and amputation. In diabetes, the glucose that is absorbed from the bloodstream after a meal stays in the bloodstream. Normally, this glucose is absorbed by the cells of the body and used in aerobic metabolism to produce ATP. Insulin is a hormone that signals the cells in the body to absorb the glucose from the bloodstream. In diabetics, the sugar stays in the bloodstream, and this essentially starves the cells of their energy source. So a diabetic cells have to resort to using other molecules as an energy source for aerobic respiration, which can lead to a buildup of byproducts called acid ketones or keto acids, which can lead to a condition called ketoacidosis. The production of keto acids in conjunction with the elevated levels of blood glucose can lead to a host of physiological problems, including blurred vision, possibly leading to blindness, fatigue, kidney problems, and frequent urination, which can lead to dehydration, possibly eventually leading to a need for dialysis, loss of sensation in the limbs, especially the hands and feet, poor wound healing, increased risk for foot lesions, which could possibly lead to amputation, a weak immune system, impaired cognitive function, irregular heartbeat, possibly leading to a heart attack, coma, or even death. So basically, in diabetes, even though you're eating and getting glucose into your blood, the glucose is not getting from the blood to your cells, and your cells are essentially starving. So number one, cells require glucose as a source of energy for aerobic respiration, also called metabolism. Briefly describe the 3 steps that are involved in the production of ATP within a cell. Where do each of these 3 steps take place in the cell, and what is required for this process to occur? So I'm going to put up a little diagram here. So we're going to talk about things that happen in the cytoplasm versus things that happen in the mitochondria. So step 1 is glycolysis. Glycolysis takes place in the cytoplasm. It does require glucose, and glucose is converted to a molecule called pyruvate. So we're not going into all the detail that's possible. We're just doing a summary. Step 2, the pyruvate goes into the mitochondria to the citric acid cycle. So this part takes place in the mitochondria. It indirectly requires oxygen, so oxygen has to be present for this to happen. And pyruvate is broken down into carbon dioxide, which is a waste product, and we use our respiratory system to obtain oxygen and get rid of the carbon dioxide. And then step 3 is the electron transport chain, which also takes place in the mitochondria. This part directly requires the oxygen, and then the electrons that were released in these first 2 steps are used to make ATP. So notice all of this starts with glucose, which is a sugar. And so with diabetes, the glucose stays out in the blood and it doesn't come into the cell so that you can have this reactions, right? So the cytoplasmic reactions are on the inside of the cell. The mitochondria is on the inside of the cell. So in order to make ATP, glucose has to get inside the cell. Number 2, describe the structure of ATP. How does ATP act as an energy molecule? Explain how the body stores energy in ATP, and then how it releases the energy that was stored in ATP. So ATP is similar in structure to a nucleotide. It has 3 parts. It has a ribose sugar, which is a pentose sugar, meaning it has 5 carbons. It has an adenine, which is a nitrogenous base, and it has 3 phosphate groups. And ATP stands for adenosine triphosphate. So the phosphate groups, particularly the 3rd and the 2nd ones, so these 2 on the end, are attached by high energy bonds. So moving these phosphates from one molecule to another is a method of transferring energy. So if we have extra energy and we want to store it, we do that by adding a phosphate group to ADP. So if we have a lot of extra energy laying around, we would take ADP, we would take that energy and use it to stick this phosphate on, which would give us ATP. Then when we need the energy, we release it by removing the phosphate. So now we take the ATP, we rip the phosphate off, that releases energy, and it leaves us with ADP. Number 3, in type 1 diabetes, the body is unable to manufacture the hormone insulin. Without insulin, cells do not receive the signal to take up glucose from the blood. Insulin is a protein, and we have learned about protein synthesis in this chapter. Explain the pathway for insulin production, beginning with the DNA and ending with the exocytosis of insulin from the cell. Use the terminology you have learned in this chapter. So I'm going to cover up part of the question up here just with a picture in the cell of this is actually called the endomembrane pathway. And this has all the different steps for making a protein. All right, so we're going to make a protein. In this case, it's going to be insulin. So first, we start in the nucleus. DNA in the nucleus contains the information to make insulin in what is called a gene. The gene is transcribed from DNA into mRNA. The mRNA undergoes modifications, like the introns are removed, the exons are fused together, and then it's finally going to leave the nucleus. All right, so now we've left the nucleus. The mRNA is going to attach to a ribosome that will attach to the rough ER, so it becomes a bound ribosome. So that's the ribosomes you see stuck to the rough ER. The ribosome translates the mRNA code to make the protein insulin, and then the insulin is going to be inserted into the rough ER as it's made. So the rough ER is going to make some modifications to it. It's going to pack it into a vesicle, and it's going to ship it off to the Golgi apparatus. The Golgi apparatus is going to further make modifications to the protein, so the final steps to turn it into the final insulin protein. The Golgi is going to package it into another vesicle and send it up to the cell surface. And then the vesicle will fuse with the plasma membrane in the process called exocytosis, and it will release insulin to the outside where it will be released into the bloodstream. So in type 1 diabetes, the insulin cannot be made by the body because the cells that normally do this process, the beta cells of the pancreas, have been killed usually by an autoimmune disorder. So the body has attacked its own beta cells, and so you no longer have the cells that make insulin. Now in type 2 diabetes, the body can make insulin, but the cells become unresponsive to the insulin signal. So discuss how glucose gets transported into a cell using what you've learned about cellular transport. Hint, the concentration of glucose is higher outside the cell than inside the cell. How does insulin regulate this process? All right, so we have a picture here. So we're going to pretend the little green molecules are glucose. So we just said that outside the cell, we have a higher concentration of glucose than inside the cell. Glucose is big enough that it actually requires a carrier protein to get across the plasma membrane. And because we're going from high to low, this is a process called diffusion. We do not need energy because we're going from an area of high concentration to an area of low concentration. And because we need a carrier protein, it's called facilitated diffusion. And again, this is a passive process that does not need any energy. So in a healthy person, so in this picture over here, glucose is still represented by the little teal dots, and insulin is represented by the purple triangles. So in a healthy individual, insulin binds to receptors, and again, going back and remembering, receptors are specific for the ligand that binds to them. And when insulin binds to the receptors, that is a signal for the cell to put the glucose carriers out and let them work, right? So then the glucose carriers will work to bring glucose into the cell. With type 2 diabetes, the person can still make insulin, but their cell has stopped responding to the signal. So the insulin can bind to the receptors, but it's almost as if the receptors are deaf and they don't hear the message, right? So they don't do anything, which means that the glucose carriers are never put into the membrane, and then they are never able to transport glucose. And the reason why the insulin stops, or the receptors stop responding to the insulin, is due to a whole lot of factors that are still under a lot of research, but it has to do with what's called a metabolic syndrome. It typically happens when people are obese, they don't exercise enough. There are a lot of lifestyle choices that can lead to these signals just ignoring the message. But the bottom line is that insulin is required for glucose to get into the cell. In type 1 diabetes, the insulin signal is not there. And in type 2 diabetes, the insulin signal is ignored. And you'll cover this again next semester. Number 5, diabetes is often linked with high blood pressure. Let's think about this critically using what we have learned in this chapter. If sugar, a solute, stays in the blood and does not get taken up by the cells of the body, how could we describe the osmotic concentration of the blood as compared to the cells? Since glucose is unable to cross the plasma membrane to get into cells, water could move instead by osmosis. What direction would the water move? How could this cause high blood pressure? All right, so let's think about this. So here's a blood vessel, and it's full of glucose because we've just eaten a nice meal, something sweet, sugary. We got lots of glucose in here. And then we got our poor starving cell down here that doesn't have any glucose. All right, so normally in a normal person, you'd have insulin that tells the cell to start taking in the glucose and everything would be good. But in the diabetic, the glucose stays in the blood cell and the cells starve. So how would this affect water movement? Remember, water, or let's compare these two solutions first. So the blood vessel would be hypertonic compared to the cell. You could also say that the cell is hypotonic compared to the blood vessel. Bottom line, we are saying the blood vessel has more solute than the cell. And remember, water wants to move toward an area of higher solute. So water would want to move from the cells and the tissues into the blood vessel. So if you add a lot of water from the body cells and tissues into the blood vessel, you are increasing the volume of blood inside the blood vessel, and that increases blood pressure. So that is one of the reasons why diabetes is linked with high blood pressure. Part 4 is an HPV case study. And sorry again that chapter 3 has such a long activity, but it is a long chapter, and I wanted to have an activity or questions that covered the different parts of the chapter. So human papillomavirus, or HPV, is a virus that is transmitted by sex. Evidence has recently shown that women who have HPV are at increased risk for cervical cancer. HPV infection is also found in about half of all penile cancers. HPV infection has also been linked to cancers of the vagina and anus. Further evidence suggests that HPV may even be responsible for certain mouth and throat cancers, as it is spread by oral sex as well, and I have more information about that at the end. HPV is expected to replace tobacco as the main causative agent for oral cancer in the U.S. We will discuss a typical cervical cell, how it could be affected by HPV, and what makes a cell cancerous. So number 1, consider a currently non-dividing cervical cell. What stage of the cell cycle would this type of cell be found in? Well, we know that it's not one of those cells that never divide. So cervical cells are cells that line the cervix, and linings or epithelial layers always divide. So we know that the cell is going to divide eventually, but if it's currently not dividing, it would be in the G1 phase of interphase. Now consider that the cervical cell has received a signal that it is time for it to divide. Describe the stages of the cell cycle that the cell would go through as it prepares to divide and then actually divides. Make sure to differentiate between mitosis and cytokinesis. So it was hanging out in the G1 phase. Next, it would go to the S phase. Remember, S stands for synthesis. So this is where the DNA is replicated, or you can think of new DNA is synthesized. From the S phase, it would go to the G2 phase, which are the final growth preparations, so the last minute preparations before dividing. And so G1, S, and G2 are all part of interphase. Then it's going to go through mitosis, and mitosis is the division of the chromosomes and the nucleus. So it divides the nucleus and the genetic material. And then cytokinesis is the final stage, and that's where you divide the cytoplasm and the organelles. And now at this point, you would have 2 complete separate daughter cells. Viruses are very small particles composed of protein and nucleic acid, either RNA or DNA. When a virus infects a cell, its nucleic acid is inserted into the DNA of the host cell. This foreign nucleic acid contains information to make a protein, so it contains its own genes, but it's inserting those genes into our DNA. In the case of HPV, it inserts genes that produce proteins that disrupt the cell cycle and interfere with cell cycle control mechanisms. What do we call these type of genes? What happens when the cell cycle is no longer controlled? So in a human that hasn't been infected with a virus, when one of our genes that normally controls our cell cycle mutates, we call those oncogenes. And in this case, the virus is actually inserting genes that disrupt the cell cycle. They would still be called oncogenes, but it's just we get them in a different way. And that's why this virus is so dangerous, because this virus is inserting genes into our DNA that disrupt our cell cycle. An uncontrolled cell cycle can lead to uncontrolled cell growth, which is cancer. Number 4, most of the time when the HPV genes cause increased cell growth, the resulting growth is benign. For example, HPV causes genital warts. Low-risk strains of HPV tend to only cause benign warts. What does it mean for a growth to be benign? So a benign growth remains in the tissue where it forms. So in this case, we're talking about the epithelial tissue of the genitals. It does not get out of control enough to disrupt normal functions. So it stays within the tissue that it forms and it doesn't disrupt normal function. But occasionally, the uncontrolled cell growth can lead to further mutations that can cause a growth to become malignant. High-risk strains of HPV tend to cause cancer. What does it mean for a growth to be malignant? Discuss using the terminology you learned in this chapter. So malignant growths have gained the ability to spread into surrounding tissues. So if we use this image here, the primary tumor is the original site of the cancer growth. And so in this case, we're talking about the genital epithelia. That's where the HPV is usually going to infect the cells and insert its genes. Then the cells invade surrounding tissues, so that's called invasion. When the abnormal cells reach the lymphatic or circulatory system, and they're showing a blood vessel here, they can then spread to distant tissues and organs throughout the body. Secondary tumors will start to grow in other areas, and at this point the cancer is said to have metastasized. So they can also call these metastatic tumors. Also, a little side note, what's interesting about cancer cells too, if you remember, we mentioned in the lecture video that cell division and growth takes a lot of energy, right? You have to have a lot of glucose, a lot of ATP. And so cancer cells have the ability to cause new blood vessels to grow to supply them with the extra nutrients they need to keep growing. And that's a process called angiogenesis, where a tumor calls a brand new blood vessel to form and essentially to feed it. And so that's another active area of cancer research. If we can somehow keep tumors from causing these blood vessels to grow, maybe we can starve them. So here's my last little public service announcement. It's been known for a while that HPV is linked with both cervical cancer and penile cancer spread via penetrative sex. But there is now increasing awareness that this virus can also attack tissues in the mouth and throat. It can spread by oral sex. I mentioned it at the beginning because when I first made this case study several years ago, it was known, but it wasn't widely known. Now it's getting more and more widely known, and I see more and more news articles about it online. HPV is in fact now the cause of about 12% of all cancers, which is kind of scary. There are 14 million new HPV infections every year. So what can you do? There is an HPV vaccine because it is a virus, so you can be vaccinated against it. You can also get oral cancer screenings at your dentist. And if you have anything that shows up in your mouth, any unusual sores, patches, or other abnormalities, an area that suddenly gets rough, something that's just not right in your mouth or throat, get it checked out immediately. And that is the end of the Chapter 3 activity.